Hey everybody, it's Mr. Poller. In this video, I'm going to walk you through the modeling meiosis activity for AP Biology Unit 5. The goal of this activity is to model the process of meiosis so that we learn about the connections of genes and chromosomes and how traits are inherited. In this model, we are looking at chromosomes 1, 7, 9, 11, 15, 16, and then the allosomes, which are the X and Y chromosomes. On each of these chromosomes, we're usually looking at one gene. So on chromosome 1, we're looking at the RHD gene, which controls if blood type is positive or negative. On the allosomes, we're looking at a gene called OPN1LW. This is the gene associated with seeing red color. Your first step is that you will be doing an evaluation of a set of chromosomes for one parent. You'll either get a handout for parent number 1, or you'll get a handout for parent number 2. Your task is to look at the chromosomes and to determine which alleles that individual has to determine their genotype, or which combination of alleles do they have, and then their phenotype, which would be the expressed trait. After we've analyzed the chromosomes, now we're going to be moving into the modeling part of the activity. I'm going to arrange the chromosomes in order of length. Okay, so I've got 2 copies of chromosome 1, 2 copies of chromosome 7, 2 copies of chromosome 9, 2 copies of 11, 2 copies of 15, 2 copies of 16, and 2 allosomes. In this case, there is an X and a Y chromosome. This combination of chromosomes, remember our model does not include all 23 pairs of chromosomes. We're only looking at 7. Because we have 2 copies of each chromosome number, 2 copies of chromosome 1, 2 copies of chromosome 2, the cell that these chromosomes would be in would be called a diploid cell. We're going to be modeling the process of meiosis described in class. So one group is going to be modeling meiosis in the parent that has an X and a Y chromosome. The other group will be modeling meiosis in the parent that has 2 X chromosomes. Leading into meiosis, all of these chromosomes are going to become duplicated. So I need to copy all of my chromosomes. So I need to take another handout and I need to do some more cutting. So now I'm going to arrange all the chromosomes so that they're matched up with their duplicate copy. So I'm matching 9P with 9P. Here I'm matching 9M with 9M. So the M and P stand for maternal or paternal chromosomes, because each and every one of us in our 23 pairs of chromosomes, we received 1 copy of that chromosome number from our maternal side, so from the egg, and we received 1 copy of that chromosome from our paternal side of our family, or from the sperm.
Okay, so now we're ready to continue our simulation. I am going to arrange my chromosomes now into tetrads. So when I arrange the tetrad, I have my copies of chromosome 1 here. There are the 1P. There's 2 of those. Those are the 2 copies of the paternal chromosome 1. And then I have the 1M, the 2 copies of the maternal chromosome 1. Just doing all my tetrads. Here I have all of my tetrads. The next part of the simulation will model the process of independent assortment. So the maternal and paternal chromosomes are going to get divided into the 2 cells that are formed at the end of meiosis 1. And this is a random process. So I've brought in a coin to do a coin flip. So I actually found a Maryland coin, so I could use my home state coin for this activity. Okay, I'm going to do a coin flip for each of the tetrads, okay? And whatever result I get, I'm going to move those chromosomes to this side of the table, and the other chromosomes, the other 2, will come towards me. So I'm going to flip first for chromosome number 1. Okay, here we go. Ready? Okay, I got tails for chromosome number 1. So that means I am going to move the 1P chromosomes this way, because they're also labeled with the letter T. So that's for tails. And I'm moving the H copies of chromosome 1 this direction. So this is sorting these chromosomes. Now I'm going to flip for chromosome number 7. Tails again. Okay, so I'm moving the tails chromosomes towards you. I'm moving the heads chromosomes towards me. So the first coin flip when I got a tails didn't make me get a tails again. That is an independent event. Now I'll flip for chromosome number 9. Here we go. I got a heads. So I'm moving the heads chromosomes towards you. I'm moving the tails towards me. Now we flip for chromosome 11. And I got a heads. So 11 H's go towards you, 11 T chromosomes go towards me. Chromosome 15, tails. So the 15 T chromosomes come this way, 15 H towards me. 2 more flips. I got heads. And then for the allosomes, heads. Okay. After completion of the cell division, we now have 2 cells that have these 2 groups of chromosomes. Now, these groups of chromosomes are going to be divided again in meiosis 2. So I'm going to simulate that by moving this group of chromosomes into 2 piles. I'll put one here and one here, and I'll do the same thing with these chromosomes towards the back, right? So I've got a 1 to the left and to the right, 7, and these are the same. So it doesn't really, I don't need to randomize this. They were already randomized. Okay, so there's my 2 groups. Same thing is happening back here. 1 and 1, 7 and 7, 9 and 9. Here goes 11 and 11, 15 and 15, 16 and 16, and the sex chromosomes or the Y chromosomes are going this way. Because we were looking at the parent who had an X and a Y chromosome, this is the parent whose body would produce sperm cells. So these 4 piles now would represent the formation of 4 cells. And these cells would now be haploid because they only have half the genetic information the original cell in our model had. The final step is to determine which of these collections of chromosomes will contribute genetic material to the zygote. A zygote forms when a sperm fertilizes an egg. So I'm going to randomly pick one of these piles by doing a very cheap spinner. Okay, so I just have a paper clip and a pen. I'm going to set it down here, and I'm just going to spin the paper clip, and whichever pile it points to, that will be the sperm cell that is going to fertilize the egg of my partner group. Okay, and my spinner is pointing to this pile right here. I'm going to discard those other chromosomes. We'll take this sperm cell and use it to fertilize an egg cell, and then we'll determine the genotype and phenotype of the child that would result from that cross.